Welcome to the Alain Elkann interviews, an unprecedented window into the minds of some of the most well-known and respected figures of the last 25 years.
So today we speak to Andrea Califano, who is the Clyde and Helen Professor of Chemical System Biology at Columbia University, professor. He completed his doctorate studies in physics at the University of Florence in Italy, and then a postgraduate studies at MIT, and he worked for IBM for a long time. And then later, around 2000, he went to work or to teach at Columbia University. Recently, he is the chairman, the president of the Chan Zuckerberg Biohub in New York, which is part of other hubs that belong to the Zuckerberg family, and they research and cure, prevent. And the purpose, if I understand well, is to manage and prevent all kinds of diseases by the end of this century, 10 to 15 years. Is this true? Is this what you do? It's a joint venture, if I am not wrong, of three very important universities. One is the Columbia, obviously, University. The other is the Rockefeller. And the third one is the Yale University, where you combine different kinds of scientists, engineers, and all sorts of researchers. And you are the boss of a team, right? If I understood.
Yeah. Just to give you a little bit of a sense of how the biohubs work, they're typically organizations that are funded around three institutions. So there's one in San Francisco, one in Chicago, one in New York. And so each one of those cities is really connecting three main institutions and then a number of satellite institutions that can also participate. And I think overall, if you take the 9 institutions that are now associated with biohubs, they represent about 65% of the overall NIH budget distributed to universities. So these are really some of the top research centers in the United States.
Mark Zuckerberg, who I think is financing a large part of this project, which is, by the way, I think shared. I don't know if it's shared financially, but in partnership with the city of New York, I guess, the state and the government. Zuckerberg, what he wants is create a new tool or platform that all scientists can use to study and make more specific advances.
Yes.
This is, I think, the goal of this.
I think it's quite remarkable because the Zuckerbergs have decided to essentially give away about 98% of what they own, money that they made, to just completely not-for-profit endeavors based on advancing science and education. Really, the goal of these biohubs is to create a novel funding model for pursuing science, a funding model that allows scientists to dedicate themselves 100% to doing the scientific research and not to continuously trying to raise funds through grants. As you know, especially in the United States, in Europe is a little bit different, but in the United States, almost all of the research that is done is essentially funded through a mechanism of filing grants, proposals, and then waiting until they get funded. And about 15% of them, 10%, depending on the pay line, get funded. So there's a lot of applications that are actually written by even very well-known or very productive scientists who don't get funded. And so what Mark and Priscilla are trying to do is to create an environment where, rather than focusing on continuously writing proposals and trying to get funded to do the next piece of science you want to do, you already know that you have about 10 years and likely much longer than that to be able to focus to create a dream team, which is another thing that is very difficult to do with current mechanisms for funding, to solve a specific problem. So think about it as putting a man on the moon. You don't do it by having a single lab working on it. You really need a community to be formed around it. And for instance, as an example, the kind of things that we're trying to do, re-engineering the immune system, this is something that in little pieces and elements is done in some labs, in some, 4 more labs around the world. But there's no example other than this biohub of a concerted effort to do it, where you create a team of scientists that work together to try and solve that problem.
How did you put together your team? Why there are engineers, physics? You're not a doctor yourself, and probably there are also researchers and doctors. How did you put together your team? And which qualifications do these people need in order to pursue this project?
So I have a passion for building teams. And this has been, when I was at IBM Research, I started and created the first IBM Center for Computational Biology. And then I started 3 companies, which of course requires creating teams and bringing people together. And at Columbia, I really founded a completely new department dedicated to system biology, which again is about bringing people together. And I'm a very strong believer in cross-disciplinary activities in biology. So even though I'm trained as a physicist originally, for the last 30 years, essentially, I've been working in biology. So I'm a practicing biologist. My lab is half dry, which is computational, and half wet, which means we do the experiments. So we really love to bring together people. So as a result of that, you end up knowing pretty much everybody in the world that does these similar things. And so putting together the team was not a huge challenge because the 3 universities you just named, Columbia, Yale, and Rockefeller, really have stellar people, and they were all super excited about participating in this endeavor.
One of your major goals is harnessing the immune system, right? And so can you please explain what does it mean, this? I mean, you think that it's not always good to use medicine, right? It's good to prevent the illness, or at least to know that there is an illness coming and then detect it and eventually try to cure later. Now, there are many other places in the world where they do all sorts of scanners and tests and things that try to prevent. But if I understand, you say that very often this comes once a person is ill. You have a symptom or you have something, a sign, and then you go and then they find out, and sometimes it's in time, sometimes it's too late.
Yes, you covered up pretty much exactly what the problem is. Just to give you a sense, for instance, the time a cancer is typically detected, it already has about 1 billion cells, right? Wouldn't it be wonderful if we could actually detect cancer when it has like 100 cells? Because if you could do that, then you would be able to treat it in extremely simple fashion without requiring either complex surgeries or complex treatments that are often ineffective when the tumor has grown too large, especially when it's progressed to metastatic disease. Similarly, there are now drugs that can help alleviate some of the progression and symptoms of both Alzheimer's and Parkinson, but they are very ineffective if you use them when the disease is already in full-fledged flare. Even in that case, wouldn't it be wonderful if we could actually start administering those drugs at the moment the disease is just in its initial phases of progression? So the immune system is the natural defense about pretty much anything that can go wrong with our body. I mean, not many people realize it, but when you actually cut yourself, the major system in our body that takes care of that is actually the immune system for 2 reasons. One, it participates in what's called wound healing. That is, it repairs your own tissue, but also it prevents foreign pathogens from getting into your body. And so it prevents bacteria from getting in and kills them on the spot. And so this is the only set of cells in the human body that can pretty much go anywhere. It can go to every single...
What are the immune cells vis-a-vis the others?
So the immune cells are cells that derive from our bone marrow. They're derived from our, what is called the hematopoietic system, which is basically the system that generates all the blood cells. And they are basically divided in 2 different classes of cell. One is called lymphoid, which includes the T cell and the B cell. Those are the cells that actually kill foreign organs. There are also other type of subset of those cells that do other things, that regulating, for instance, inflammation. And then there are myeloid cells that also participate in many other functions. So there's a very complex system with literally 100 different cell types, each one with a very specific role, and each, in types that are very well defined biologically. So what we're trying to do is basically to take over where natural evolution has stopped. Natural evolution has basically shaped our immune system to defeat most of the challenges that we encounter before we enter our reproductive age. The immune system is based on the law of reproduction. So if a species can reproduce effectively, what happens after reproduction is no longer very important because it's no longer participating as a shaping force in modeling the organism. But what happens before the reproductive age is actually what evolution shapes our organisms to do. And so we, for instance, probably get cancer 1,000,000 times a day. It's just that the moment a cancer cell emerges in our body, our immune system takes care of it, because otherwise we will not be able to survive long enough to have progenies. The problem is that it also takes a very long time to fix the genetics that give us certain type of functional traits in a population, about 200,000 years. And so you have to think about what reproductive age for the human species was about 200,000 years ago, maybe somewhere between 15 and 20 years old. So the reality is that basically anything that happens after we're about 20 years old is not something that our immune system has been shaped to defend against. It has been shaped to defend against the thing that happened before that. So, as I said, wound healing, infectious diseases, those were the major killers of humanity. And those are the things that the immune system knows how to do.
How do you use them? I think your goal is to try to convey these immune cells to fight like they do before you're 20 years old.
So the way the immune cells defend against an infection, for instance, which they're trained to do, is by detecting certain type of signals or messages on the surface of the bacteria or viruses, and then basically creating an inflammatory response so that they recruit all the other immune cells to that site, and basically they participate in killing the foreign object, right? What they're not trained to do yet is basically the ability to sense other type of signals that are emitted by cells that, for instance, are associated with Parkinson or Alzheimer's or cancer, the type of cancer that are able to evade the initial immune response. And so what we're trying to do is to engineer these cells so that they can now recognize the signals, which they are not trained to recognize yet, and then activate exactly the same mechanism that they could have used in order to kill a bacteria in the case of cancer, or actually prevent inflammation in the case of diseases that are autoinflammatory or neurodegenerative. And so basically, it's all about teaching the immune cell how to get to a particular sites where there is a problem in one of our organs. And for doing that, we're actually designing novel technologies that allow us, for instance, to find out every single cell in the human body where an immune cell gets in contact with, right? And once you know that, now you can figure out how to read the behavior of that cell and figure out whether it's doing the right thing or it's doing something abnormal. So this is basically how we are using some relatively simple form of engineering. We're not creating a Frankenstein. We're simply taking something that has already been done, for instance, to fight cancer. You've probably heard of something called CAR T therapy. CAR T therapy basically modifies an immune cells so that it can recognize an antigen that is on a cancer cell. And once you do that, then now the T cells is able to kill the cancer cell. We're doing the same thing, but instead of using a mechanism that can recognize the antigen of a cancer cell, we're using something that could recognize the antigen or a signal emitted by an Alzheimer's cells or a Parkinson cells, et cetera. And so we're just generalizing a concept that has already been used in cancer to essentially do much more generalizable things.
And you do this with the help of artificial intelligence or just by yourself?
That's a great question. So a lot of what has been done in immunology so far has been essentially by intuition and careful assembling of clues that can then be used to formulate hypotheses. So it's a process that is quite complex and is very much trial and error and to some extent serendipitous. So for everything that we discovered in immunology, there's 100 things that did not work out. What we're trying to do is trying to turn the laws of that discovery process to our advantage so that when we go to the lab and we formulate a hypothesis, the probability of success, the probability that that hypothesis will be validated is around 80%. And how do you do that? You do that by essentially using computational models based on machine learning or AI that can actually simulate or model what the cell is going to do when it encounters a certain stimulus, for instance, or is perturbed by a drug, for instance, so that once you have that model, it can actually predict the solution to the problem you're trying to solve. So for instance, let me give an example. There are some cells in our immune system called regulatory T cells. And what they do is to keep inflammation at bay. That is, they basically tell, for instance, our gut, don't get inflamed. We know that you're seeing a lot of bacteria, but most of these are actually harmless. You should not start going completely out of control and start inflammation. So these are called regulatory T cells. They block inflammation. Cancer recruits these cells. So it fools the immune system into thinking that it's actually okay and it should not get inflamed and therefore attacked by the immune system. So what you can do is by changing a single gene that we could find computationally, we basically were able to prevent the regulatory T cells to go to the tumor sites. And when you do that, the tumor goes into spontaneous remission. You don't even need to treat it with a drug. So this is, I think, a very powerful example of the kind of things of how we're using computation. We basically use computational models to ask the question, what is the gene? What is the gene that controls the ability of the regulatory T cell to go to the tumor? And now when it's blocked that gene, they can no longer go there. How many do we have to test? We only had to test 17 genes. And of those, about half of them were experimentally validated. So it becomes a very powerful tool because there's 20,000 genes, and it would be almost impossible to do an experiment where you try each one of those 20,000 genes.
I read that you think that, I don't know when, but all this will be detected easily by a blood test instead of machines and all that.
That's a goal. The goal is to create what we call a cellular endoscope. An endoscope is essentially a only minimally invasive device that allows you to go inside our body with a minimal incision and basically to detect if there's anything wrong. So, for instance, a lot of times you can do it to look at your stomach or to look at your intestine, etc. But there are certain places where you can't go. You can't go to the brain. You can't go to your pancreas with an endoscope. But if you had a cellular endoscope where certain cells are instructed to go to a specific site and to report back on what they've seen, now you'll be able to do very, very complex diagnostics. The idea is that we're creating cells that could actually be reinjected into the human body and be able to go to any organ on demand. So the design cells to go specifically to the pancreas, specifically to the brain, etc. And then report back what they've actually seen there, whether there's anything abnormal. And then they would be naturally killed by the human immune system. They will simply live a little bit longer than what is required in order to report back. But then after about, say, a week, they would die. And the cells would be cells essentially from your own body or cells that are compatible with your body. So there wouldn't be cells that can potentially become cancerous or anything dangerous for the body. So that's the ultimate goal that we're trying to have. And that will probably be reached in the next 10 years. For the time being, what we're trying to do is to figure out what are all the different pieces of this puzzle that we have to put together to reach that point.
Why one has cancer, why one has Alzheimer, why one has Parkinson, where are these coming from? Because we know that if I cut my finger, I make a cut, and then eventually, or if I stay naked in the cold, I will probably catch a pneumonia, something like that. But before knowing if there will be a penicillin or another kind of medicine who will kill the cancers, why do they come? And why one has Parkinson, another one has Alzheimer? Do you know that?
Yeah, so cancer is simply the price that we pay for being an organism that is still capable of evolving. So our cells, when they double, they accumulate small variants, called mutations, that are actually responsible for allowing the species to further, specialize and potentially generate even additional species down the line. So the price that you pay is that sometimes some of these mutations, even though there are extraordinary mechanisms in place that prevent almost any cancer, because otherwise we get cancer every time a cell divides, sometimes these mechanisms are bypassed by mutation that co-occur in very complex ways and creating a pattern that defies the internal mechanism of defense of the cell. And then in that case, that cells become cancers. Now, you're still a long way off from getting cancer because that's exactly the point where the immune system can actually detect that something went wrong and will kill those cells. But because of these mutations, some cancer cells have now evolved. So think of cancer being evolution inside your body. These cancer cells have evolved to develop mechanisms that allow them to keep the immune system at bay. It basically either, for instance, recruits regulatory T cells or they recruit other type of cells that basically tell the immune system to stay away. And now the cancer can start growing. And once it grows beyond a certain size, then it kind of becomes an extra organism inside our body. So that's why we get cancer. For Alzheimer's and Parkinson, there are very similar mechanisms. The complete causal etiology of Alzheimer's is not yet fully understood, but we know that there are problems that have to do with the formation of fibers. Proteins essentially are the main building blocks of the cell. And sometimes these proteins can start forming fibers where they aggregate with each other. And when they do that, we still don't know whether that's a consequence of the Alzheimer's onstart or whether it's actually the cause of the actual disease. But in some ways, these cells are very abnormal because they have this protein called tau fibers that are very damaging to the cells. They don't allow it to do the proper function they normally would have. In Parkinson, you have similar type of disease. In other diseases, like, for instance, ALS, this is amyotrophic lateral sclerosis, you have the motor neuron, which are essentially the longest cells in our body that connect our brain to our peripheral musculature, get essentially killed. So you start killing them because the specific signals that you're sending them essentially tell them to kill themselves. And when that happens, all of a sudden you start losing control of your peripheral musculature, eventually of your lung, ability to breathe, etc. So you end up very rapidly degenerating and being unable to control your body. So these are all things that are routed partly in the genetics of our bodies. So there are certain variants. For instance, there's a variance in the apolipoprotein E that is associated with the increased risk of Alzheimer's. And similar, for instance, there's a protein called SOD1, superoxide dismutase 1, that is 100% predictive of whether you will get Lou Gehrig's disease, it's called ALS, by the age of 30. So if you have that mutation in your germline genetics, you're guaranteed to get to that disease. Similar, there are proteins that will tell you you're guaranteed to get Huntington disease. So essentially written in our genetics. So other things are not so clear. For instance, there are a lot of forms of Lou Gehrig's disease that are actually not yet associated with a specific genetic insult. And so they're probably a function of both genetics and of the environment. What you were exposed to, maybe initially there was an hypothesis about fertilizers because soccer players were ending up getting more likely to get ALS than other people. So it's a very complex conundrum that combines both the genetics of our body, the mutation that we accumulate because of errors in when the cell produce, and of course, the environment.
You said that to be in New York for your center is important, not only because New York is a very important center of universities, research, and all that, but also because it's very multi-ethnical. Not every population has the same problems, or it's the same.
You're absolutely right in the sense that typically the studies that were done until about, I would say, 30 years ago, were studies that were mostly done in white male populations. For instance, there were almost no children that were enrolled in these studies. Many of them did not enroll many women, or at least that is a significant imbalance. And certainly they did not enroll many of the minorities. And then about 30 years ago, we started to realize that the ability of disease to manifest in humans is very much depending on the background genetics of our bodies. And those are obviously very strongly linked to the ethnicity. So the genetics of, for instance, an African American in the United States, it's quite different from the genetics of, say, an American of Asian descent or American of European descent. In Africa, there is probably the largest diversity, even within one continent. In fact, mapping the genealogy of Africa has been one of the major efforts that have gone on in the last 30 years. African American women tend to be much more likely to get breast cancer that as a more lethal form of breast cancer than white women. And so there has to be a careful investigation into why there are these discrepancies, these differences. That can only happen if you can do studies in populations that are multi-ethnic and really represent the way.
The Biohub will have one way of proceeding for this kind of trying to govern, let's say, the cells.
Immune cell?
Yeah, is good for everyone. Process of detection, is it the same for all the world, or it changes according to what you just said?
So that's exactly the point. So what you want to know is, what is the most universal mechanism of detection, right? If you studied only, say, African-American population, or you studied only a white American population, you may find things that you can detect that are very useful, but they may not carry across to the other ethnicity, right? If you actually study them all together, you can distill what is the most generalizable principle that will help, right? Because of the partnership with the three universities, we will have access to very large populations. And so by studying those populations, we'll make sure that the mechanisms that we try to target are the most universal one we can get.
By harnessing the immune system, you think that one day, I don't know, 10, 15, 20, I don't know how many years, cancer, Alzheimer, or Parkinson will be like a flu, that you get it and you will get rid of them, or not?
I think for cancer, that's definitely a very strong possibility. For Alzheimer's and Parkinson, I think what we are certainly hoping to accomplish is to be able to detect them at a stage where they're still treatable. treatable in terms of really delaying the full onset of the disease. Once that's what is settled, then there's no drugs to invert the progression of Alzheimer's.
Tell me something. These illnesses, right, which not always, because sometimes they touch very young people or even children, all that, but they always existed, and it's just now.
The problem is that we live a lot longer now. For instance, cancers has always existed. You also have cancer in children, unfortunately. So a lot of the work that we do, including in my lab, is actually on pediatric malignancies. But they're very, very rare. So that typically in children, the incidence of cancer is a very, very small fraction of what the incidence would be.
Instead, it's very bigger and bigger when you get older, right?
Correct. And so basically, that's one of the reasons why the cancer that you get in older people is not something that the immune system has been trained to see, because you're past your reproductive age. And we're starting to see a lot more cancer because people live longer. So if people died when they're 20, you would see very little cancer. Now we're 85 average, and people live, Rita Levi-Montalcini, I think it was like 105 when she passed away. And there are people that have been living until 130. So I'm just saying, when you live that long, then the probability that some of the cells in your body may go awry.
What does it mean, doctor, that a human being is not programmed to live too long unless artificially helped?
No, we're just programmed to live just as long as it takes to get children. That's what we're programmed to do. Everything else is a bonus. It's extra time, as you would say, right? So that's the way natural evolution works.
Okay, Professor, thank you very much for your participation. Now we have David on the other side.
Yes, thank you. One question I had is, two questions actually. One is do you see an observable effect from what I would call late generation? Now you talk about people reaching reproductive age. Now people are delaying having children until much later in life. Is there an observable effect that you see at a cellular level from this delay?
There's a famous experiment. You know what is the best way to prolong the life of flies? You prevent them from reproducing until they're old, and then they live longer. So I think it will take a long time for whatever genetic event that will actually increase our lifespan to actually take hold in the population. But we're certain, in terms of evolutionary processes, the later you have your children, the more the natural evolution is putting pressure on your genetics to live longer. The question is, can we have children when we're 90 years old? That would probably be a difficult solution. But for instance, I think we've already seen, I'm not an expert in that field, but I think we've already seen a significant increase in the age at which women can actually be in a reproductive age and still be fertile. And I think that potentially is a consequence of society evolving towards not having people reproduce when they're in their early teens, which is what used to happen probably 200,000 years ago. So that definitely puts a pressure on the race living long.
And unfortunately, I have a friend who had cancer recently, and his oncologist said to him, well, now it's 1 in 2 people who get cancer, but in 5 years' time, it's going to be 1 in 1. That was his opinion. Do you see that kind of increase? Is that simply because people are living longer, or is that because of other effects? And how are you going to be able to...
In terms of actually etiology of the disease at the same age, it is going down in most cancers, especially due to smoke cessation and to better eating habits, except for GI tract cancer. Gastrointestinal tract cancer are surprisingly increasing. So stomach, colon, and small intestine. The reason they're increasing is probably because we eat a lot more meat, a lot more burned meat. And so our diet has gotten significantly worse. And that certainly has an incidence on the population. They're still rare. These are not super common cancer like prostate cancer. I would say probably when you say every person will have cancer, every man will have prostate cancer. There's no question about it. I think we all already have prostate cancer. It's just that you have to determine what are the cancer that will kill you and what are the cancer that will not kill you, right? So for instance, one example is that in breast cancer, due to mammographic diagnosis, you now catch an enormous number of cancer that you would not have been able to see before. But they're not cancer. They're called in situ carcinoma, and they may never progress to be a challenge to the life of the individual, except that now when you see them, you take them off, because obviously nobody wants to live with a potentially a time-ticking bomb in their body. But if you didn't know you had them, probably 60, 70% of those would never progress to an actual life-threatening cancer. So when you say that we're all getting cancer, I would say yes, but it's also becoming a lot more treatable and a lot more preventable at the same time. But the question is, how many people die of cancer? That's a real statistics you have to look at.
How do you see the treatment options for people? Is this going to be very expensive and very personalized, or will it be quite general?
So the initial way in which this will work is called an autologous transplant. That is extremely difficult to implement. What autologous means, it means you take the cell from your own body, you engineer them, and then you put them back, right? Obviously, you can imagine that requires time, it's very expensive. It's like creating a medicine just for you. That's not the way we see this working long term. So there are certain mechanisms that allow us to create cells that are universal. Basically, what is the big problem? The problem is that if I take my immune cells and I inject them into you, they will die instantaneously. The immune system will recognize them as not being cells, and they will be immediately wiped out. But there are some mechanisms. For instance, there is a particular signal on the surface of the cell called CD47, which is also very famous, called the don't eat me signal, that basically tells cells that they should not be eaten because they're not yet expressing, it's called the HLA, that is the receptor that allows other cells to recognize them. And so, for instance, you could express that signals and a couple of other signals to basically prevent recognition by the immune system for a short amount of time. And in that case, you now can create a universal population. So you could see a day in which you go to a doctor and they have a set of vials, right? And each vial contains a different type of cells. You know, you have a cell that goes to the stomach, you have a cell that goes to the pancreas, a cell that goes to the brain. And then basically, depending on what diagnosis you want to do on that day, or maybe even a mixture of all of them, you can simply inject them into any person, and you will no longer need a personalized approach to using these type of ways. Now, this is one of the reasons why we have so much emphasis on the basic science of immunology, is because we need to learn more about this kind of mechanisms so that we can harness them as well to generate universal therapies. So the first therapies we'll generate will probably be personalized, but then the same exact therapies will be generalizable so that they can be used in anybody.
Okay, thank you very much.
Alain Elkann interviews.